So good afternoon, everyone. As mentioned, my name is Renata. I'm from University of Macau, China. And today I'll present the work titled Enhanced Removal of BTEX, DCE, and cis-DCE mixture using waste scrap tires immobilized with indigenous Pseudomonas species. As the topic of this conference is related to recycling, our group proposed to use scrap tires to enhance the removal of some volatile organic compounds. But what's the purpose to use scrap tires? Well, around 3 billion scrap tires are discarded annually. And according to the United States Environmental Protection Agency, in the end of 2013, the United States generated around 209 million scrap tires. And this waste material has been mainly used as fuel. Recycled products are exported to developing countries as retreated tires. Here we are talking about the largest scrap tires recycling market. But even with all the recycling and reuse efforts, among 1/4 of scrap tires end up in landfills each year. Landfilling scrap tires actually is another environmental problem due to the leakage of toxic components, organic and inorganic ones, during tires decomposition. So under these circumstances, new utilization of scrap tires to reduce the amount of waste is more than welcome. Tires have a complex composition that depends on the utilization application. Here we have an example of car tires composition, and the main ones are carbon black and rubber. These two components can exhibit remarkable adsorption properties as activate carbon, a well-known adsorbent used in environmental application. But the utilization of scrap tires as an adsorbent is quite new. Here we have a study from 2014 that utilizes scrap tires in a two-phase partition bioreactor to remove a mixture of derived phenols. The obtained results are very promising for tire utilization, especially due to the increase of 2,4-dichlorophenol adsorption on tires and consequently reduce the toxicity to bacteria. And also, we have an improvement in the removal efficiency of both contaminants, using 10 to 15% of tires per weight. So in our study, we work with two different kinds of volatile organic compounds. The first one is BTEX. BTEX is a mixture of contaminants that includes benzene, toluene, 2-benzene, and xylene isomers. These contaminants are the most major monoaromatic compounds present in most petroleum products. And we can also find BTEX application as an industrial solvent and equipment cleansing. BTEX are also listed among the most priority pollutants due to the acute and long-term toxicity, especially in case of benzene, known as carcinogenic. One of the most common sources of BTEX contamination in soil and groundwater are here spills from leaking oil tanks. The second group of compounds that we choose are chlorinated aliphatic hydrocarbons, most known as CAH. They are the major compounds that we can find in subsurface due to the long utilization as a cleaning and degreasing solvent. The trichloroethene, I think, is the one of CAH that are most studied. Is the most frequent found contaminants in groundwater and exhibit higher carcinogenic properties and toxicity. So up to now, there is no microbe that can be isolated from environmental samples that can use CAH as a carbon source. So in biological process, CAH can be removed from contaminated area by two kinds of mechanisms depends on the redox condition of the site. The first one is under an aerobic process. So here I give an example. TCE can be reduced all the way to a thin. But this reaction is often incomplete, and we can observe the accumulation of byproducts that are even more toxic to TCE. The second way is anaerobic. In this case, TCE can be fortuitous oxidized in the presence of organic substrate, can be BTEX, to produce harmless products in a process that we call co-metabolism. Currently, BTEX and CAH, they can be removed from contaminated sites using different kinds of technologies, biological, chemical, and physical. Attention has been paid to the biological technologies due to their cost-effective, environmental-friendly nature and the possibility to produce harmless products by mineralization. Although the co-occurrence of contamination is getting more often, we have only few studies relate to the simultaneous removal of both contaminants. Further improvements still need to be done, especially in the removal of CAH by microorganisms, which is very poor. So we justify the development of hybrid technologies we employ in biological together with physical or chemical technologies. So the objective of this work is to develop a hybrid technology utilizing scrap tires to enhance the bioremoval of CAH and BTEX, utilizing microorganisms, in this case, Pseudomonas plecoccocicida. The experiments were divided in two groups. The first group was biological study, and the second group was adsorption study. In these two kinds of study, we evaluate the utilization of scrap tires. For the biological study, we prepare two different kinds of microcosms. The first one we call suspended system. We call suspended system, that we have only microorganisms with the solution. And the other system was immobilized, that we have scrap tires together with microorganisms and the contaminants. The microcosms were prepared in certain bottom, containing mineral salt medium, the contaminants in this concentration. In the case of BTEX, the total concentration was based on the mass fraction of all the components present in crude oil. In this case, the bacteria suspension and different amounts of scrap tires. The bottles were incubated for 5 days under these optimum experimental conditions, and the liquid phase concentration for all the contaminants were checked daily by gas chromatography with flame ionization detection. For the adsorption studies, we utilize the concentration of all the contaminants the same as the biological studies using tires, and we get sample in different times to check the concentration of all the contaminants to finally calculate the adsorption capacity of all the contaminants to evaluate the utilization of tires as adsorbent. In this study, we use motorcycle scrap tires. They were cut into small pieces, washed and with deionized water and dry in an oven. The amount necessary for biological and adsorption studies were weighted and autoclaved before use. The microbial culture, we first isolated from a heavily contaminated site near a gas station in Xiamen, China, and then we further identified as Pseudomonas plecoccocicida. This strain has a high capacity to degrade BTEX, and we use this strain to also co-metabolize CAH. The absorption capacity of the contaminants, here we can see that was highly dependent on the contact time, reaching a maximum in about 1 day. Toluene was the most sorbent, followed by benzene, xylenes, ethylbenzene, and CAH. This order does not follow any particular chemical property, but the concentration of all the contaminants present in solution. Then we further evaluated the adsorption capacity when all the contaminants were present at the same concentration, in this case, 100 milligram per liter. So we prepare one bottle containing all the contaminants in the same concentration, and we name as multi-solute BTEX. And then we prepare different bottles with all the contaminants present alone in each bottle. And then we call single solute BTEX. Under this condition, the adsorption capacity follow another order. Ethylbenzene and xylenes were sorbed more, followed by toluene and benzene. This order followed the octanol water partition coefficient for all the contaminants, suggesting the hydrophobicity of tire surface of the sites. The removal efficiency were also calculated for all the contaminants in single and multi-solution, and we observed that no significant difference between these solutions, suggesting that there is no competitive adsorption between BTEX components for the adsorption sites on tire surfaces. For the biological study, we calculated the removal efficiency for all the contaminants after 5 days of incubation under different conditions. So here we have only tires, here different amount of tires. Here we have the immobilized system. Means that we have microorganisms tires along with contaminants. And the third one is the suspended system. You have only microorganisms and the contaminants. So for the suspended system, we can see that Pseudomonas can degrade well BTEX, while the removal efficiency of CAH is very poor. Here, substrate limitation is a common concern because BTEX and CAH, they share the same enzymatic pathway. But if after we add tires to the system, you can see improvement in the removal efficiency. But here, the improvement was most by adsorption of CAH on tire surfaces. When we work with immobilized system, the highest removal efficiency for all the contaminants were obtained when 4 grams of tires were provided. And for the suspended, only to check the adsorption, they also follow the same order of adsorption. Xylenes was most sorbed, ethylbenzene, toluene, benzene, and CAH. The hydrophobicity of tire surface was checked with contact angle experiments. And here the value is higher than 90, which suggests tires has a hydrophobic surface. Pseudomonas species are known to produce a hydrophobic polymer to attach on hydrophobic surface. And we could imagine that tires should be ideal surfaces. But after we count the viable cells that were present in the solution and on tire surface, we evaluate that 20 to 25% of the microorganisms were attached on the surface. This actually is a low adsorption of Pseudomonas, and improvements still need to be done, especially if we want to improve the removal efficiency, the biological one, that is more important for the CAH. So now we are trying to modify the tire surface in order to create a more hydrophobic environment for the adsorption of microorganisms. So the conclusion is the scrap tires is considered a good candidate to enhance removal of volatile organic compounds, such as BTEX and CAH from liquid phase, due to the remarkable disruption properties as well as the capability to immobilize microorganisms. Further studies are in progress to evaluate whether the immobilized microorganisms possess the same initial removal efficiency as reutilization cycles, including whether higher concentrations of the contaminants can also be applied. So this is my presentation. Thank you so much.
Thank you, Ms. Alves. Is there any question? No questions. I have one comment, because this is not my field of research, but is there any other product or any other element to remove the BTEX? Or
For example, by absorption, we commonly use for the removal of BTEX, we can find some other technologies, chemical or physical technologies. Physical using activated carbon. But in this case, we have the problem, but we are only transferring the contaminants from one medium to another. Biological process, we have chemical process, we have lots of, but they are very costly. And for the biological, we also have different kind of strains of microorganisms that can use BTEX as carbon source. But in case of CAH, we also have chemical and physical and biological process. Mm-hmm.